Well, good afternoon, everyone. Welcome to our IGB lecture today. We have a special pioneer lecture today, and I'm very pleased and honored to introduce our speaker, Stephen Quake, who has a pioneer lecturer, who's the Lee Otterson Professor of Bioengineering, Professor of Applied Physics at Stanford University. Professor Quake earned his Bachelor's of Science degree in Physics and Master's degree in Mathematics from Stanford in 1991. He received a Doctor's of Philosophy from Oxford University as a Marshall Scholar in 1994. He went back to Stanford to do postdoctoral work in the laboratory of Nobel laureate Stephen Chu on single molecule biophysics. He started his independent academic career at Caltech, quickly rose through the ranks, ultimately was appointed the Thomas and Doris Everhart Professor of Applied Physics, and then moved back to Stanford in 2005 to help launch a new department of bioengineering. Professor Quake also is a very successful entrepreneur, has founded or co-founded several very important successful companies, including Fluidigm, Helicos Biosciences, Verinata Health, Quanticell Pharmaceuticals, Moleculo, Cellular Research, and Immumetrics. He, since 2016, served as co-president of the Chan Zuckerberg Biohub in San Francisco. And the Chan Zuckerberg Biohub is one of the largest programs in the Chan Zuckerberg Initiative. And most recently, he's serving as president of the CZ Biohub Network. In July, he took on still another role and became CZI's new head of science, overseeing the entire philanthropic portfolio in the Chan Zuckerberg Initiative. Professor Quake is renowned for numerous paradigm-changing tools in biophysics, genomics, and biotechnology. He's invented many tools to measure biology, many of the tools that many of us use routinely, especially at the single molecule and single cell levels, including game-changing DNA sequencing and cell isolation technologies. The Quake Lab has also used these innovations to advance biomedical science with non-invasive blood tests to detect premature birth, Down syndrome, and organ transplant rejection. As you can imagine, Professor Quake has received many awards. I'll list a few of them: the Human Frontiers of Science Nakasone Prize, Jacob Heskel Gabay Award, the MIT Lemelson Prize for Innovation, the Raymond and Beverly Sackler International Prize in Biophysics, an NIH Director's Pioneer Award, numerous fellowships in a variety of professional societies. He's also a member of the National Academy of Sciences, National Academy of Engineering, National Academy of Medicine, one of less than 24 people on the planet with that trifecta, and also is a member of the National Academy of Inventors. Please join me in welcoming Professor Quake to the podium. Here is a little memento, not so little, memento of your visit here. Don't worry, we will ship it to you. And I don't know if you can tell what single-cell transcriptome that is, but it's not a quiz if you can't.
I'm not off the top of my head.
There you go. Okay, all right.
We can talk about it later.
That's right,
Yeah.
exactly. Please.
All right, thanks so much, Gene. It's great to be here. I'm going to talk to you about the work we've done about cell atlases over the last decade or so, but want to talk a bit about the history. And there's a long history of cell atlases in biology. You can trace it back to any number of things. Here I've listed a few prominent ones: Ramon E. Cajal's optical microscopy of the brain, all the beautiful pictures of the cell types, their morphologies. Don Fawcett's book, An Atlas of the Cell, using electron microscopy to take pictures of many different cell types and their organelles and constituent parts and so forth. Then moving into lineage maps of Sulston and Horvitz, their work with C. elegans. Sidney Brenner, 20 years ago, made a proposal to make something called the cell map. And this was in his Nobel Prize acceptance lecture, so you can go look it up. And in his inimitable way, he had his finger on the pulse of the whole thing, that, the human genome drafts had just been published, and, he pointed out that the job is not done by any means, because for any multicellular organism, the genome is not really the blueprint. That's the wrong analogy to use. Every cell in the body more or less has the same genome, but they specialize in quite different cell types. Neuron is very different than a lung cell, different than a muscle cell, and so forth, despite having the same genome. So the genome should really be thought of as a parts list, and every cell type uses different parts from that parts list. And having the genome doesn't tell you how any of that works. You need to figure that all out. And so he proposed another project to do that called the cell map. And that's sort of essentially what we've ended up doing as a community, making these molecular cell atlases using the tools of single-cell transcriptomics. And that in itself, the technological developments of that can be traced back 3 decades easily. And these are some of the highlights along the way to my mind. Almost as soon as PCR was invented as a biochemical amplifier, people began to think about how to use it to amplify genes from single cells. And in 1990, there were 2 papers. Jim Eberwein and James Isco published papers showing how to make single-cell cDNA libraries through amplification. But they didn't know how to read it out. You didn't have the technologies to read stuff out in parallel in those days. And so they were using southern blots. And they'd look at 1 or 2 genes to show, hey, we made a single-cell cDNA library. And figuring out how to read out the sort of really sort of complex multiplicity of information there took many more years. And in 2003, 4 groups published papers showing how to read out single-cell cDNA libraries using microarrays and hybridization. And some of these were done on homemade microarrays, some were done on commercial microarrays, ranged from hundreds to thousands, and the commercial ones, tens of thousands of genes you could read out in parallel. And so that kind of really established how to read out the information in these libraries in a way that was scalable and approaching sort of the true complexity of what's there. And then, of course, people moved from hybridization to sequencing to read out the information. And the first demonstration of that was in 2006 with Morose and Kendall, where they were using Sanger sequencing, probably one of the last big experiments with Sanger sequencing, to read out single-cell cDNA libraries from C-slug neurons and thousands of transcripts from single cells to show that that could be done. And then a few years later, Fuchu Tang used next-generation sequencing to read it out. And this kind of put the field on a pretty good footing in the sense that you could make single-cell cDNA libraries, you could read out the information in them, but the remaining challenge was how to do it at scale. Because all of these were sort of bespoke experiments done on 1 or 2, a handful of cells at a time. And what was missing was, we'll let you do this on lots of cells. And that's sort of when I got involved in the field, was in helping to solve that problem. And my group for many years have been working on developing microfluidic tools to automate biology, developing the fluidic equivalent of the integrated circuit instead of having transistors and logic gates, had valves and pumps and things like that, did tens of thousands, ultimately hundreds of thousands of valves on a chip, and use that to explore a number of different questions in biology and biochemistry and cell biology. As Gene mentioned, I founded a company called Fluidigm to commercialize that, and they had done that at scale. Eventually, worked out that they had produced and sold more than 1 billion valves, which is more than all the conventional plumbing in, Texas, California, and New York combined. And they created the first commercial instrument to do single-cell genomics. It was called the C1, and it was based on both the founding the core technology and work we'd done in my lab to build chips to isolate and amplify cDNA. And this allowed one to do 96 cells at a time. This sort of opened the whole field up. Hundreds of labs around the world began using this instrument and allowed people to sort of, for the first time, do these single-cell experiments at scale and take apart complex systems. And it eventually got supplanted by a second generation of technology. Funny enough, based on work we had done 2 decades earlier, making microfluidic emulsions on a chip, as an alternative way to do sort of microfluidic experiments at scale. And a group in Boston, led by Dave Waits and Steve McCarroll and Aviv Regev and Mark Kirshner, eventually turned that technology into a different tool for doing single-cell genomics based on capturing cells and beads and droplets. And that got called various things, DropSeq, InDrop, whatnot, eventually commercialized by a few companies, most successfully by 10x Genomics, and that provided another couple orders of magnitude of scaling and throughput. And we use and continue to use both in my lab, super powerful, but now all the pieces are in place for anybody to do this, and it's quite widely used. And so I call the talk a decade of molecular cell atlases because lots of interesting ideas about doing this for whole organisms were floating around in 2011 and 2012. We had been doing work sort of one tissue, one tumor at a time at Stanford, and realized we should be doing this for the whole organism. And so I wrote a proposal called a cell type atlas for mice and humans. At the same time, more or less, Sarah Teichmann was moving from Cambridge to the Sanger Institute. In her job interview with Mike Stratton, she said, we should do a human body map using these techniques. And that launched a big effort at the Sanger. Also around that time, Aviv Regev was launching the Klarman Cell Observatory at MIT. It was initially focused on understanding cell circuits using single-cell techniques, but very quickly pivoted to making atlases. And over in Sweden, Israel, Sten Linnerson and Udi Shapiro wrote a really nice essay suggesting also it's time for a systematic characterization of cell types across organisms using these single-cell transcriptomic techniques. And I sort of tried to collect this whole history in the essay published earlier this year. But it was all one of these things, several of us in different corners of the world all sort of thought this is the time to do it. We can take these nascent techniques and apply them at scale to really fully characterize all the cell types of an organism. That was 10 years ago. Now it's basically done, certainly at the level of rough draft. And I'll take you through some of the stories of the pieces that I was involved in. What we've done in the Bay Area is create whole organism cell atlases of mouse, fly, lemur, and human. In the case of mouse, look at it across several different perturbations, including aging and rejuvenation. And these were really big collaborative efforts. Team science, these papers have 150, 160 authors on them, involve dozens of groups from, in the case of mouse, lemur, human, Stanford, UCSF, Berkeley, and the Biohub. In the case of fly, we did that as an international consortium that involved people all over the world. And sort of really fun to do and get done, and not the sort of science I had been doing in my career up to that point. And part of what it took to get it done was to create a new research institute called the Biohub, which we were able to do with the generosity of Mark Zuckerberg and Priscilla Chan. That provided us kind of the oomph to do this and kind of the central resources. We set it up as an independent research institute, but one that was closely collaborating with Stanford, UCSF, and Berkeley, and try to take on projects they wouldn't do on their own. And the Cell Atlas ended up being sort of a great example of one of those. So let me tell you a bit about some of what we did along the way. We started with mouse, and this came about from a conversation with my colleague Tony Whiskeret, who had been collaborating with for years and who's interested in aging. And he said, we have some real aged mice in our lab, and we should do some transcriptomic characterization of them. I said, well, let's do single-cell transcriptomic. We just started the Biohub. We're gearing up for this, and that launched the whole thing. And what was very important about that conversation was realizing that if we're going to do aging as something we were interested in, there's not a lot of mice. It's hard to get them to live a long time. And we ended up, our oldest mice, 30 months old. That's like the equivalent of a centenarian for humans. So we knew that we wouldn't be able to have a lot of animals to do this on. So we spent a lot of time figuring out how to collect as many tissues as we could from any single organism and coordinating all the groups we got involved to do this as a big team project. So we sacrificed a mouse. Every morning in the summer of 2016, we sacrificed a mouse in Tony's lab, and there was this group of students and postdocs waiting with their ice buckets outside to take their tissue back to their lab to do the dissociation, sorting, and whatnot, and then bring it all back to the Biohub to have it sequenced. And so this idea of recruiting a bunch of tissue experts, labs that were specialized in each of the organs, having them participate, coordinating all the collection, and then doing the sequencing and informatics of the Biohub as a central facility kind of became the keystone for how we did a number of these projects. And working out all that teamwork and logistics became essential to it all. So a couple of the key people involved, Spiros Dermanis, who's a young group leader at the time at the Biohub, and Angela Pisco, who's in data science at the Biohub, helped pull it all together. And I'll introduce you a bunch of other folks along the way. But what we ended up doing was the first paper was just the 3-month time point. We called that the tabula muris. We did something like 18 different tissues from each mouse. And then we did the aging study, ranging from 1 month up to 30 months. That became the Tabula muracenis, the aging mouse atlas. And I had to consult with one of our professors of classics at Stanford to make sure we got the Latin right for that. And a lot of argument, a lot of opinions about what Tabula muris meant. That was fun to work out. So those are the first 2 papers. And these, I mentioned several of the postdocs and students involved. At the bottom, some of the folks at the Biohub who are doing the sequencing, led on that side by Norma Neff, who runs the genomics facility, and Jim Carcanius, who had data science. And so we, for each single cell, sequenced the transcriptome, 500,000 cells in the atlas, and then could ask questions. So each cell becomes essentially a 20,000 dimensional vector, each dimension being one of the possible genes, and you're understanding at what level, or if at all, it's expressed in that cell. And you can ask questions of the data set, like questions that don't involve any biology, like how different are these transcriptomes? Just put it in the computer and ask each of these 20,000 dimensional vectors, do they cluster differentially in that space? And you can try to map it down to lower dimensions to see if they do, and you get all these clusters and structure out of it. And several things pop out of that. They're sort of interesting. Number one, these are colored according to tissue of origin. So you can see most of the clusters are different single colors, and so the computer's figuring out, oh yeah, these cells are different from different tissues. We get that. Don't need to know any biology. It's enough to know the expression. Some of them are multicolored, and those turn out to be cell types that are shared across tissues. Things like endothelial cells, things like B cells, and so forth, things like T cells. And so computers are also smart enough to figure out, just based on the transcriptome information, that in fact there are commonly shared features in cell types shared across tissues, and it identifies those. And here you see all the names superimposed on top. Where do those come from? Well, that came from our recruiting of the tissue experts. So besides doing the experiment, after each tissue was sequenced and subclustered, they went through and identified each cluster and gave them a name based on the differential gene expression pattern. And that kind of human expert annotation made this one of the largest expert annotated data sets in the literature, and it had been downloaded many, many times because of that. And every new algorithm, people try to benchmark it against this because of the high degree and quality of annotation of the cells within it. That also became something we ended up putting a lot of effort into in all the other projects we did, making sure that we had good expert annotation. So, what else? Lots of interesting questions one can ask about what defines the cell types. In this case, asking which transcription factors are contributing to cell type definition uniquely, and you could do things like calculate Gini coefficients and get lists of transcription factors that define them, and lo and behold, many of these were known to be important in reprogramming. And so we essentially have is now a recipe book for how to go about trying to find new reprogramming protocols for different cell types, pretty much any cell type you want, based on all the ones that weren't known. And so here, for hepatocytes, skeletal muscle, oligodendrocytes, the colored ones were ones that were known in the literature to be useful for reprogramming. We now have other candidates to explore, as well as many for other cell types. I mentioned we looked at making atlases across ages. Lots of interesting questions to ask there. Here's one example, senescence. So cells expressing P16 or CDKN2A, which is sort of senescent marker. You can see that the number of cells expressing it increases with time. And so the number of senescent cells increases with time, and the intensity of expression increases with time. And then looking at these cells, we identified many other genes that are involved in senescence and sort of teased apart. Interesting story there. You can also look at mutations. By looking at mutations in the transcripts, we can look at mutational load over time. Without getting into too many of the technical details, included NIS traceable RNA controls in many of the experiments. And so those are defined synthetic transcripts where we know they shouldn't be changing with age. That's like a baseline for this estimate of mutations. That's the non-shaded curve, and you can see that across cell types, that kind of defines the technical lower limit of measurement for sensitivity to discovering mutations. And then you can see that as you move from 18 to 24 months, mutational burden is increasing similarly across cell types. And many other interesting things came out. I won't dwell on it. It's published. You can have a look at the paper. I'll tell you a bit about the third and final chapter of the mouse story, which was to this was one of our initial motivations, try to see if we could reverse aging with parabiosis. So this is something that folks like Tony and Tom Rando have been working on for years, where if you take two mice and surgically attach their vasculature together, they share a common blood supply. Parabiosis means living together. And in the context of aging, they had been doing parabiosis with young and old mice together. So you hook together old mouse, young mouse. They share the same blood, and the older mouse, by some physiological measures, appears to get rejuvenated, and the younger mouse appears to get older, accelerated aging. And we thought, all right, we can now explore this at a real molecular level. And to be honest, I thought we were going to be able to call bullshit on the whole field. I mean, I thought it was probably all artifact, and this is going to be a big null result, and we'll show that all this parabiosis stuff is nonsense. But in fact, it actually turned out to be quite the opposite. Super interesting, and there is really interesting molecular things going on. This is we've done so many of these. I can't keep them all straight. I didn't recognize the one in your thing. The shapes are a little funny the way these get elongated, which is a bit of a clue, but yeah, don't totally have it off the top of my head. So this is sort of one of the interesting examples of how to look at atlas data all at once. What you have here are each column is a different gene, each row is a different cell type. So if you want to ask a question about genes whose expression change with age, you can do that, but you can ask it not just for one tissue, you can ask it or one cell type, you can do it for all the cell types and ask, is this a consistent phenomenon you're seeing universally across many different cell types? That's part of the power. So here, what we're doing is we're looking at genes that change with age, and we're asking, is it consistent with the natural aging measurements we made on the tabula muris senis? And so each little circle there at the intersection is color-coded, and the left half of the circle is the result from the accelerated aging. The right half is from the rejuvenation, so you flip the sign. And we're asking, is it consistent or not consistent with natural aging? And so blue is consistent, red is inconsistent. And what you see as you look across this broadly, the chart on the left is genes that are downregulated with age, chart on the right, upregulated with age. The ones that are downregulated with age, largely consistent in the parabiosis model. It actually does make sense. And so when they're rejuvenated, they're going the other way. When they're accelerated, they're going faster in that direction of aging. And, it kind of hangs together. The ones that are upregulated, less so. Those are more artifacts of parabiosis, a lot more red on that side. And so give the flavor of how you can tease all this apart at a real molecular level. What's sort of the most single takeaway here, the most consistent thing we see across all the data cell types? Genes from ATP synthesis, electron transport. And the mitochondria, aging, that's really what we see. Downregulated with age, gets reversed, and it's very, very consistent across a large number of cell types across many tissues. I'll tell you a bit about fly. So after we published the first mouse paper, the fly community approached us, asked for help making a fly atlas. They were feeling left out because people were doing human, mouse, They wanted to do a fly atlas. And we said, sure, we'll help out. We're not going to do it for you, but we'll teach you how we did it, and we'll help do it. And we'll do a certain amount of the sequencing and whatnot. And so this was a great, as I mentioned, international collaboration involving Bart de Planck in Switzerland, Stijn Ertz in the Netherlands, Norbert Perelman in Boston, Brian Oliver at the NIH, and a bunch of other folks. We kind of started out doing something very similar to what we had done with the mouse, which was do individual tissues. Not all from the same fly. Don't need to do that with flies. There'd be no trouble getting lots of flies. But we definitely want to do individual tissues. So recruited different tissue expert collaborators. They would do dissections, pull out their tissue. We do the single cell measurements on each tissue individually, and put all the data together and annotate it with Jamboree. Now, with all the other atlases, we were using live cells. We dissociate the cells, use live cells, and do those measurements. And typically, we did two kinds of measurement on each one, so we'd have a chance to understand the technical errors that are being introduced by either 10x or SmartSeq. With fly, we decided to use nuclei because the fly chitin armor is hard to dissociate and just harder to get all the cell types. And so we decided we'd sacrifice a little sensitivity by doing nuclei. That was one difference. And then went and sort of this is it. Here a bunch of the tissues. You can see again for each tissue, beautiful separation of cell types. They've all been given names by experts. Many of these had names before, but only 1 or 2 genes were known. Now, thousands of genes defining each cell type. And so a really super kind of reference companion for Drosophila. And the thing that I love about insects now, and I'm really hot on this for some of my research going forward, what's very special about insects is it's trivial to make a cell atlas because you can put the whole thing into the 10x lane. You don't actually need to go and do all the different tissues separately. And so we did experiments where we did a whole head or a whole body and put all the nuclei in and ran them, and you recover almost all the cell types. So it becomes very, very straightforward to build these cell atlases, and you can now march through every important mutation and every important mutant line and say, well, what's the effect of this mutation across the physiology of all the tissues in the insect? And I think that's going to be super fruitful going forward, and very special about these organisms. Maybe true for bees, too. Here's another couple examples of what we got on the fly atlas. So here, nice analysis of the transcription factors again. And, there are 2/3 of them here on this graph, so 500 out of the 700. Each column is a different cell type. And as you see, all these transcription factors, more or less, each cell type is defined by a transcription factor. And so all of a sudden, we get a bunch of things out of this. Number one, we know what this transcription factor does in terms of defining cell type. And number two, we know where to go to look now to understand which genes it's regulating and chase down the actual regulatory network. And so, you're getting a big swath of the regulatory network of the organism all in one go here. There's some funny ones there in the germline cells. We won't talk about those right now, but that's another interesting one on its own. Another interesting thing about flies is looking at sex-specific gene expression. So turns out there's this gene called DSX where it's ubiquitously expressed across all cell types and tissues, but there's isoform differences for male and female. And so we can then sort of post hoc go and figure out for every single nucleus in the data set, did it come from a male or a female fly by looking at the DSX isoform. And so you can see in the green, it's ubiquitously expressed across the cell types. Now you can look at which genes are biased by gender by looking at that isotype. And there's clear bias, right? Sexes are not equal in how they're doing gene expression. And if you look at the right here, you can say, which cell types appear to be the most biased by sex, and it's a bunch of ones for the intestinal tract in the female, and other ones, fat bodies and pterocytes in the males, in interesting ways, adipocytes rather. Sorry. So lots of things to dig through, and with all these atlases, we've just kind of scratched the surface. I mean, the work is in making these giant data sets. They're all shared with the community. We can tell a few of the stories in the first papers we publish, but there's much, much more to do over time with them, and they'll be revealing their secrets for a decade to come. So I'll talk a bit about human as well. This is one that we've been sort of building up to, and we ended up taking the same approach that we did with mouse in the sense of trying to get as many tissues and organs as possible from any given donor, with the idea that would be holding a lot of things constant that you want to hold constant. Holds the genome genetic background constant, holds age, environmental exposure, epigenetics, all that would be held constant. So that's the approach we took. I'll tell you a bit about how we got there in a few minutes. Just want to introduce some of the folks here. Bob Jones, who sort of quarterbacked the whole thing in my lab, senior research scientist, and the number of faculty members from all the institutions involved: Julia Salzman, Nir Yosef, Mark Krasnow, Angela Pisco, Jim Kirkinius, Norman Neff. The last three met from the Biohub, and the others have played important roles along the way. When we did this, we decided not just to do the transcriptomes. We also, for many of the tissues, had professional pathologists look at it and do the histopathology, and all those images are included as part of the data resource. And we also, for a couple of the donors, were able to measure the microbiome in the colon through sectioning and direct sequencing of 16S RNA. So we've published the first draft earlier this year, and you can see how it breaks down across donors. We had 4 donors where we got a large number of tissues all from the same donor, and you can see that it was kind of increasing over time as we were getting better at doing it. And so each column there is a different donor, rows the different tissues. And then we had another 11 donors where we were getting 1, 2, or 3 organs from them along the way at various points in time. And so those are shown in the final column, and those provide biological replicates and all sorts of other important aspects of the measurements. And so totals up to 15 donors, 500,000 cells roughly. We'll continue to do the work. We took another donor 2 weekends ago, actually. Got our biggest number ever, 21 tissues from one donor. And we're now well up over 1 million cells and thinking about what the next iteration of the release, data release is going to look like. So how do we get these donors? We're partnered with an outfit called Donor Network West. They're a nonprofit organ procurement organization. They're the ones who help find people for doing organ transplants. And when you give your body to science, they're the ones that get called in. And Donor Network West serves Northern California and Nevada, about 13 million people. They've been absolutely terrific. They, first use of all these tissues is for transplantation. If they're not going to be transplanted, they're used for scientific research, and we get a shot at it. And they've just been absolutely awesome for figuring out how to do something of this complexity where you're taking, upwards of 20 tissues out of one given donor. And it requires a lot of coordination and a lot of practice. We have all our tissue expert teams do some degree of practice on single tissues. We have a giant Bible of protocols to follow when the actual donor comes in to do it. And it's obviously impossible to predict when you're going to get one. We get like a day or two's notice. There may be a donor coming. Here's the clinical file. Would you like to take this one? We have a look at it, see if it might be suitable. There's kind of a discussion among our group. And if we decide to go forward on the day of, then it becomes sort of an all-night fire drill. It seems to always happen at night. It takes about 24 to 36 hours of nonstop work once it starts coming, because the surgeons work in the operating room one tissue at a time. And our folks are waiting outside with the ice buckets and couriers, and they get sent to the various labs. And it's just sort of an awesome dedicated team that's making this happen. And here's, one donor. We got 50 10x libraries, 82 SmartSeq plates, all done in this time. Because we're working with live cells, there's a real time component to it. We're also biobanking some of the tissues as well, so I can go back with other ways to measure. But just really kind of an intricate choreography. So everything is shared on our portal, which you're free to go look at. We've tried to provide access to the data in ways that don't require you to be an expert. So there's a tool that CZI made called Cell by Gene, which makes it easy to surf through the data and look at where genes are expressed and You can also download the raw data, look at the images, so forth and so on. We found, I forget what it was, 400 cell types, 400 odd cell types. This is sort of a big tangle graph showing it. The cell types in the middle, the organs on the left, and the compartments on the right, showing just the awesome complexity of what's there. And we have, as I sort of said earlier, and as for the other atlases, a full molecular definition of all these cell types now for the first time, which is pretty amazing. There, was all this discussion in the field as people were starting various cell atlas projects. How many cell types are there? And at the second, we organized the second Human Cell Atlas meeting at Stanford. And I asked everybody, write down on your name badge, how many cell types you think there are. And somewhat disappointingly, half the people wouldn't even make a guess. The other guesses were all over the map. The textbook says 300. It went anywhere from there up to millions, up to infinity on the badges. I had made a principled calculation. My number was something like 6,000. And my principal calculation was, in 100 years or more of looking at cells under microscopes, there's no way that biologists have missed more than 95% of what's there, right? They can't have missed more than 95%. So that's the upper limit. 5% of cell types are known. So that 300 is 5%. Well, multiplied by 20, all right, there's maybe 6,000 cell types. That'd be the upper limit. And, I think, that was generous. And, the true number is less than that. Leaving the brain aside, brain is complicated, haven't worked that one out. But every other tissue other than brain, nobody's even doubled the number of cell types. And the value in this endeavor, I think, is less about finding new cell types and more about getting the full understanding of the ones that are there and the full molecular characterization of them. I feel like it's time to write a review article going back to look at all these papers that have come out about single cell this or that in this tissue. None of them has even doubled the number of cell types. They always, 1 or 2 new cell types and 20 known cell types. And I think that's just the nature of what this is about at the end of the day. But what else is there? Lots of things. Julia Saltzman developed a really neat algorithm to look at splice variants in the single cell and splice isotypes. And turns out there's a ton of them there that people hadn't appreciated. We discovered hundreds of thousands of putative new splice variants and went and validated many of them in other bulk data sets where they just hadn't been apparent because it's too hard to pull out of bulk data using unbiased algorithms. And she also discovered that of well-known and well-characterized splice variants, many of them are cell type specific in dramatically different ways. And so here's an example of myosin light chain 6 across a variety of cell types using 3 different donors. And you can see that this is sort of two particular splice variants, including that exon 6 or not. The fraction of cells that use that just varies dramatically by cell type. Really noticeable, and I think unappreciated that there's going to be a huge diversity of that. And so this is, again, something we've only scratched the surface of, and it'll be super interesting going forward. These are not dynamic measurements in the sense that we're getting these donors at one point in time, but you can infer dynamic information from the data in a couple of different ways. So one is to look at cell cycling. And Geeta Mahmoodabadi at postdoc in my lab put together a nice cell cycling score to figure out where things are. Are they dividing? Are they quiescent? And we could classify each cell type according to this index and how much division appeared to be going on. It's sort of an eye chart on the left, but if you zoom in on the right, the ones that are at the top of the index, the most actively dividing ones, are ones that make sense. Various progenitor and stem cells, things in the immature enterocytes, things like that, stuff that makes sense. And so we can say that about the dynamics of the cells. You can also compute something called RNA velocity, which I won't go into here, but a beautiful sort of idea of looking at intron-exon splicing to predict where gene expression is going. And so you can, within tissues, sort of put vector fields on and say where cells are going in their gene expression space. And so here's an example from the bladder showing cells flowing from immature to more mature cells and in ways that kind of make sense biologically, which are revealing about all sorts of gene expression programs, and this can be done in essentially any tissue in the atlas. So here's some of the folks who did it. These are pictures of the folks from the mouse and the lemur. I'm not going to talk about the lemur tonight, but the preprint is up and you can read that. And again, sort of really dedicated set of people are willing to come in and in the case of human here, even larger group, work through the night when needed as we're working through these different donors. Now, we had published our paper in January and ended up publishing back-to-back papers with the Broad and the Sanger, who it turned out had also been working on whole human cell atlas measurements. And we hadn't really been talking to each other about it. And it was the editor of Science who kind of knew we were all doing it and told us, you guys ought to co-submit together. And so we did that. And we didn't talk to each other about what we were writing up, didn't coordinate on the data analysis. So we all just agreed we're going to submit our paper. We shared our abstracts and said we're going to submit on this day. We all submitted on that day. And then we all put our preprints up and kind of looked at what each other had done. And it was very interesting. We'd all taken quite different tacks on it. And you can see here a comparison of the different papers in terms of some of the scope of organs, donors, cells, and technical aspects. But the emphasis of the papers ended up being quite different. I've walked you through what we'd done about looking at trying to build a real reference where we had expert annotation, how we looked at things like splice variation, and how we looked at some pathology as well. That was our work. Broad, as you might expect from a place focused on genetics, they had really focused on the connection of the atlas with disease-causing genes and taking genes that have been discovered through GWAS and asking in which cell types are those genes expressed and where are they playing a role in the body, and that was very nice analysis there. And the Sanger folks had focused on only doing immune cells. So their atlas was 100% immune cells. They ignored everything else and did very nice work looking at sort of immune clone structure and other aspects of that and across tissues. And so it turned out to be, I think, a very nice experiment that we all found different ways to approach this problem. Now, I don't want to leave you with a sense that this is all sort of big science type stuff. These techniques are very straightforward to do at the individual lab level, and we continue to do that in my lab in ways that are quite different than the Atlas work, to ask different sorts of questions, and I'll give you a couple examples of that. I won't dwell on it because they're published, but just to point you there. So with Tom Sudoff, we've been interested in asking the question, revisiting the question, what's the role of RNA in memory, and especially long-term memory consolidation, and doing it in ways that use all the modern tools of cell biology, not just single-cell transcriptomics, but things like Leach and Lowe's TRAP2 mouse and such forth, and discovered really interesting things about what cells are doing over the course of weeks as memories are stored, consolidated, and recalled. This was work done by 2 postdocs, all right? So, and there you go. With Leach and Lowe, interested in questions of evolution of structures in the brain, particularly cerebellar nuclei. And so again, a couple of faculty members and asking, really interesting questions about what's the role of these nuclei and how they changed over evolutionary time. And now that we've made these atlases across many different organisms, there's interesting questions about how do you compare them across evolutionary time. It's really non-trivial. We know how to do it with genes. You can build phylogenies of genes and their relationships. How do you do it with entire transcriptomes of cell types? You need a whole new set of algorithms to think about that. And my colleague and your alum here, Bo Wong, spent a lot of time thinking about that and written, I think, one of the earliest and so far most important approaches to doing that, called SAMMAP, where you sort of simultaneously optimize on understanding relationships between gene homology and transcriptome relationships between species, and that's been a tool that he's used very productively to compare things like fish and frog, and we're now using in many different aspects as we try to understand relationships between these atlases. And this just scratched the surface. There's so much more interesting things to do in that I can't wait to see how that evolves over the next years. And I'm going to finish the talk by talking a bit about applications to human health and what this has to do. And we're seeing lots of people use the Tabula sapiens for things like looking for on-target toxicity by asking if you've got a drug target for something, where else is it expressed on the body, and where might it have, if you drug it, have unfortunate effects. So that's an early application that people are using. I can share a couple more mature ones that are directly involved at finding drug targets. So Mike Clark and I had been collaborating for a number of years. Mike's one of the gurus of solid tumor cancer stem cells, and we've been collaborating for a decade at Stanford, and a lot of the early development of these methods was, in my lab, working with Mike to look at tumor heterogeneity and ask, what are the different cell types within a tumor, and which are the stem cells, and what are interesting potential targets to drug within them. And so we did that academically and then founded a company called Quanticell to do that at scale. They developed 3 programs. Quanticell was acquired by Celgene, which was acquired by Bristol Myers. And now 2 of those programs are in phase 2 trials. So these are new drugs, so not repurposed. And we'll see what happens. it's, the idea was find these targets in tumor heterogeneity, develop molecules against them, and see what happens. If anyone tells you, it's not hard to make drugs, don't believe them. We're more than a decade into this, and we've not been sitting on our hands, and we still don't know if they're going to work. And this is just sort of the nature of drug development, I think. We're going to know a lot in the next couple of years, and then we'll see. And the process, here's one of them. This is the first one, BET-BRD4, where we would look at the single cell measurements here. Each row is a cell, each column is a gene. Find which are the interesting tumor generating populations, look at the targets where they're expressed, inhibit them by knocking them down, then make the small molecules. Third program is now going to go in the clinic. Hopefully it'll be 3 for 3. This one took a bit longer. We ended up out-licensing it, but IND is approved and they should go first in human, hopefully by the end of this year. This is KDM4, which is a super interesting target, especially for breast cancer. So that's one set of ways to use these tools to find new targets for drugs. Look at a complex neoplasm and find the cell population targets within that you want to drug. Another one is to find rare cell types. Another approach is to find rare cell types and have them yield their secrets. And we went about this in my lab through the study of allergies, which, as you may know, are common and sort of increasing to epidemic levels worldwide. The numbers are staggering. Something like 8% of the population is affected. And food allergies are a very specific aspect of that, which can be fatal, and sort of dominated by 8 major food allergens, but affects in the U.S. some 32 million people. Teenagers, young adults have the highest risk for fatal anaphylaxis. My daughter was one such person who had very strong peanut allergy, and it's through our experience with that I got interested in this as a health problem, and ended up collaborating, in fact, with her allergist, Kari Nadow, who runs the Sean Parker Allergy Center at Stanford and has been doing a bunch of trials for desensitization and desensitized my daughter, but current approaches have some real limitations, and we began working together to try to understand could we find better ways to do this. And it started with kind of trying to understand the cells that make the bad antibody, IgE cells. And so it turns out that these are such rare cells that they'd never been purified from humans before. So we set out to do that using these single cell approaches and succeeded in that. From patients in her clinic who were allergic, we were able to essentially separate and sequence IgE producing B cells. And this was done by my student, Derek Crute. And what we found as we looked at these different antibody sequences and looked at their relationships, that there were different individuals, patients in this cohort, who would evolve essentially the same antibody. This kind of convergent evolution has been very rare when we looked at other aspects of the immune repertoire. So here's CF1 as clonal family 1. You see there's squares and triangles there. And the shape is the identity of the patient. So you can see that clonal family made, wow, that's incredible. Something must be very important about that antibody. So we began expressing the antibodies and testing them. And sure enough, many of them are binding to the allergen, in this case to peanut. And so we realized we had potentially a platform to discover fully humanized antibodies against any substance that humans are allergic to. And that's probably useful for something. We founded a company called iGenix to try to do it at scale. They've gone and done it, and now something like 9,000 antibody sequences, a variety of different antigens, and just amazing. They went back, redid the peanut thing with a much larger cohort. Now you can see same graph as before, but many more examples of convergent evolution. The statistical confidence is way up. It's just unbelievable that this happened, but it does. So what do you do with that? Well, how does desensitization work among the conventional way of giving people small amounts of the allergen, increasing over time, making them eat a peanut every day, which is what my daughter still has to do. So it ends up somehow convincing your immune system to make the allergen-specific antibody with a different isotype, IgG in this case. So people who are desensitized have a bunch of IgG that's specific to the allergen, and that outcompetes the IgE. That's why you're desensitized. You got more of the IgG, eat a peanut, and you have some exposure to the allergen. The IgG sweeps it up, the IgE doesn't take it and go down the whole anaphylaxis pathway. And so if you could give people antibodies to the allergen that were not IgE, it could outcompete and you'd be done. And so IGeneX has taken this approach. They're re-engineering these IgE antibodies to make them to IgG and then administer them to people prophylactically. That's the theory of how you do it. So you get a shot every couple of months, and you wouldn't have to worry about exposure. You wouldn't have to eat peanut every day. You'd be protected. That's the theory. Would it work in practice? You have to find out. The company has got now the first animal data showing this works in animals, and it's super exciting that they can cure peanut allergies in mice and in rodents. And so it appears that it's going to work. It works in animals. That's a good sign for human in this case, I think, because of the way the biology matches up. And so it just, a really great result and milestone. It's going to be years before we know if it fully works in humans, because there's a lot of work to get done here and there. But I'm feeling really good about it based on the animal data. So I'll stop here and just kind of bring you back to the major theme of the talk, which is that these tools for unraveling the nature of cellular identity have become very powerful, and we've built large-scale atlases that are, you can think about them as phenotypic companions to the genome that provide molecular definitions of cell types and help interpret how the genome is used in different cell types. I didn't talk about what this has to do with physiology so much, and so there's a question about, if we're going to define these cell types by their transcriptome, how does that correspond to identity as defined conventionally by physiology? And I can give a whole other lecture on that. It's a lot of fun. We spent a bunch of effort looking at that. And it turns out, for the most part, the relationships are incredibly close. And it all makes sense in the way you hope it would, because, it's biology. It's causal. So I'll stop here. Thank you for your time, and I'm happy to take any questions.
Open for questions. Dan.
Yeah, this was beautiful. It took a while to read the science paper. And it's even better to quote your work, because if they went all the authors, your one citation takes up almost the entire page. Heavens for consortium abbreviation. But my question is, now we have this great transcriptomic information. Have you, because I do cell modeling, you don't always see a good correlation between mRNA and proteomics. Have you accompanied some of this, in some cases, with proteomic studies to help us understand the variation that we see that unexpectedly to very single cell types?
Yeah, that's a good question. So, we do have a proteomic group at the Biohub who's starting to look at some of these tissues. And, it's sort of, the field is just starting to get single cell sensitivity. And Matthias Mann has published a couple of really nice papers on that. It's this new Bruker-Timshoff mass spectrometer that's just starting to give glimmers of working. And there's other ways to do single cell protein work. Ulf Landegren had done some nice stuff with the dual antibody thing. And the proximity ligation assay. And so there's a little literature out there. We haven't gotten matching data with our data sets yet, but we have the biobank. So we can always go back and now measure as these techniques become more powerful and come online. And there's a question of why do you want to measure the proteins? And how much of the information is captured by RNA? The correlation is not perfect, but is it good enough? And from my perspective, it comes to what's the question you're trying to ask? The questions we've been trying to ask about what are the identity of cell types, I think pretty much everything useful is coming from RNA. There's many other questions about what's happening to proteins in cells post-translationally and such forth that we're not going to get out of RNA and not designed to ask in these experiments, but which I think will be interesting to look at in other contexts, and that's a whole other generation of experiments to do.
Great talk. I have a question as we start, and a lot of us here are thinking about ways to start producing engineered tissues for a range of applications, disease modeling or regenerative medicine. And at the tissue scale, we're thinking about things that are multicellular, they're hierarchical, and there's a context, usually a matrix curious if you thought about, for example, as you're starting to get a massive human data that you're accumulating about cell types, are there ways in which that data set could be informative as we think about questions of the matrix effects or donor variability, or things such as dynamic reciprocity, sort of the evolution of an environment over time? Can you use age-matched data to predict things about a regenerative solution or the like that could inform the design of tissue structures?
Yeah, I mean, for sure. I mean, this should be really useful for all of that in a lot of different contexts. And, just to come to your question of age, our median donor age is about 50. And, but it goes down. I think our youngest is 30 right now. I think they're all tragic stories, of course. And the oldest are up to 60-ish. So we're getting enough dynamic range as we accumulate more donors that it's starting to make sense that we can think about doing analyses as a function of age. And we'll probably be selecting, that'll become a criterion for donors going forward as we're trying to fill out this age dynamic range. And that'll be very interesting, I think.
As an allergy sufferer myself, I was intrigued by this last story about IgE, IgG. How does this competition really work? Does it really compete? They compete for recognition of the same antigen, or there is also some global competition? All IgEs compete with all IgGs, because I was thinking that you can somehow suppress all of the IgE repertoire by injecting some, volume of IgG.
Yeah. With the caveat that it's early days still, I will say that every antibody is specific, not just to an antigen, but an epitope on it. And, as we've delved into things like peanut, right, you can see that there are, I forget how many, 6 different bins, something like that are the major epitopes of the peanut antigens. And people will have sometimes multiple antibodies against, different bins, but they're all very specific for their own thing. And so the design of the molecules that IGeneX has been testing in the animal things covers all the major bins, okay? And so like parapitopic antibody, And so all that comes into play in making something that's going to have efficacy in the long run. Does that answer the question?
Yeah.
Yeah.
I guess this is just still work in progress, right? So
Absolutely, still work in progress. Yeah.
how allergy works.
Yeah.
As spatial transcriptomics have become more prominent, more accessible, especially at the single molecule, single cell level, how do you see that being integrated into your cell algorithm?
Yeah. Yeah. So we've been biobanking the tissues so we can go back and add these other methodologies as they come. And that's like the biggest one on our horizon right now is spatial. And we've been benchmarking a bunch of the different methods to try to figure out which ones to use. And we're leaning towards SlideSeq, which is now commercially offered by this company called Curio. That seems to be the best for untargeted measurements right now in terms of sensitivity, resolution, So we like that one a lot. We also have done a lot of work with the MERFish through VizGen's commercial thing. That's also quite good, but that's the case where you pick a gene panel. So you got to pick a set of genes and interrogate them. So within the space of having defined gene sets, we like that one. And there's a preprint out of our sort of comparisons of that. Yeah. Yeah, Rashid.
Thank you for being here.
Yeah.
You did all these different human, mouse, fly. Can you comment on, I know probably too early, but still, what can we learn, what are you learning already about the lineage and the original source of commonalities or these various cell types?
Yeah, I mean, it seems to me we ought to be able to ask questions about the origin of multicellularity from this at some level over time. As we're comparing, it's interesting to see which cell types exist in mouse, but not in human, or vice versa. And lemur is kind of the one that's in between, and it helps to understand lemur as a model organism a little bit. And there's some famous ones in the lung. So like when we got very deep in the lung with Mark Krasnow, found more than half a dozen cell types that humans had that mice didn't, and vice versa. And so you say, all right, well, is mouse a good model for thinking about therapeutics for lung and physiology in human? And it's providing a lot more nuance on that. So I think the lung is probably the most, and the lung was all done by hand. They didn't have the algorithms in and all that. And so that's probably the most carefully worked out one thus far. And if you look at our paper on the Human Lung Atlas, it's it's covered. And the Lemur paper also goes quite a bit into these comparisons. Preprint.
I'll ask the last question.
Yeah.
And that has to do with the name cell type. So in the brain, there's extensive neuroplasticity. So how comfortable are you with the concept that you can type a cell based on one state when
Yeah.
you know that the cell state can change dramatically?
Yeah, that's another good question. And, you know, it's one of these things in the early HCA human cell atlas meetings, I try to get people interested in this, and they just were just, no, let's just sequence stuff. But I think it's a really profound question. How do you find cell state versus cell type? And, you know, how do you draw the boundary between them? And I don't think as a field we have a really good definition of it. Even this notion that cell type is discrete turns out to be not true. So we've done a bunch of work on this, especially in the brain, where you can see that it came out of work we were doing again with Tom Sudoff in the striatum, that, you know, my student was looking at it and trying to cluster it, it's not quite working right, and, you know, he's saying, this seems like continuous variation of cell type. And so we went back and looked at that really carefully, and sure enough, there's a bunch of cell types where you see continuous variation. It's often correlated with spatial position, and you know, there's a whole set of cell types that have this sort of continuous spectrum rather than discrete. And you know, that's part of the whole thing right now, and I think we as a field have to do a better job kind of articulating that. You know, as to your specific question of type versus, you know, I think most people have sort of operational definition. If it's in a particular state for more than a day, it's probably can be called a type. That's more or less the best we've got right now, unfortunately.
Yeah. And Bo Wang was one of the first postdocs at the IGB Fellows at the IGB.
Yeah, there you go.
There we go. Well, let's thank our speaker for a great time.
Yep.